everyone. So, hello everyone. Today we will discuss about the polymerase chain reaction. So, in this class, firstly we will understand like what is polymerase chain reaction. So, polymerase chain reaction is a technique in molecular biology field which is used to multiply or amplify a single copy of particular DNA to produce the thousand and millions copies of particular DNA. Okay, so suppose, yeah, give me some minute. Suppose this is a kind of DNA. So when you wanted to amplify, or when you wanted to produce thousand to millions copies of this particular DNA, then the PCR, the polymerase chain reaction, will help to produce the multiple copies of this particular kind of DNA. Okay, thousand to millions copies of DNA it will produce. Okay, so it is nothing but it is the synthetic way of our replication which is happening inside our body cell. Okay, so it is the artificial way of replication of any kind of DNA, or you can say any kind of the target DNA amplification. Okay, so this PCR is based on the ability of DNA polymerase, some kind of DNA polymerase. Like inside our body also there is certain kind of polymerase which is called as the DNA polymerase, which is helpful for the amplification of DNA. Like that in the PCR also we are using some thermostable DNA polymerase to synthesize the new strand from the template DNA. Okay, so since the DNA polymerase can add nucleotide only onto the pre-existing 3' OH group, it needs the primer, okay, to which it can add the first and other nucleotides. Okay, so like in our body we have seen that while replication we require certain kind of mRNA primers, so that the DNA polymerase can add the nucleotide bases from there to amplify or to extend the particular sequence. Suppose this is the DNA, okay, template DNA inside our body, so some mRNA primer or some mRNA short sequence will come, and it will act as an initial starting point for the amplification of this particular DNA segment. Okay, it will add and the polymerase will add here the nucleotide bases, and it will extend up to where till the template is present. Okay, so like that in this case, because it is the synthetic way of the means DNA replication or DNA amplification, that's why it's require also DNA polymerase, and this DNA polymerase add nucleotide bases in the primer 3' OH in. Okay, so that it can extend the given template DNA. Okay, so in the reaction component, if you will see, there is certain things required to do the PCR. Okay, so the first one is the target DNA. Okay, so what this target DNA will do, it contain the sequence which should be amplified through the PCR reaction. Okay, so the target DNA is nothing but it is the template DNA. Okay, it is the template DNA. Okay, it is the template DNA. So it will helpful for means it is the sequence which we wanted to amplify. Okay, so that is target DNA. Then apart from that you require the pair of primers. Okay, so what is the pairs of primers? As I earlier discussed, it is the oligonucleotides, short oligonucleotides, which give the 3' OH end to extend the DNA sequence by the help of DNA polymerase. Okay, so it define the sequence to be amplified. Okay, so suppose this is your target DNA, so here it will be the or short oligonucleotide, the primer will be there, which is complementary to your target DNA sequence, and through the help of DNA polymerase, it will extend the means this complete DNA sequence. Okay, so apart from that, you require DNTPs. Okay, so this DNTPs is nothing but it is the individual nucleotide. Okay, so it is the deoxynucleotide triphosphate, or you can say like the DNA building blocks. Okay, so when you are amplifying this target DNA, for amplification you require nucleotide, so that only it will base pairing with the template and can produce the replica of that particular template. Okay, so that's why we require the DNTPs. It is the individual nucleotides. Okay, like our DATP. Okay, so it is the DATP, then DCTP, then DGTP. Okay, this is the DGTP, then TTP. Okay, so this kind of nucleotide you require. Okay, then you require one thermostable DNA polymerase. Okay, so this enzyme that catalyze the reaction. Okay, so here we are taking the thermostable DNA polymerase because here we are proceeding our reaction in a very high temperature. Okay, so for separating this DNA strand we require the very high temperature. That's why here we require the thermostable DNA enzyme, so that it can efficiently work. Okay, then apart from that you require magnesium ions. This magnesium ions is a work as a cofactor for this particular enzyme. Okay, so as we know that every enzyme giving certain kind of basal activity, and when we added the cofactor for this enzyme, it will enhance the activity of particular enzyme. Okay, so for that purpose we are adding the magnesium ion. Then we will use the buffer solution. Okay, so what is buffer solution? It will maintain the pH as well as the ionic strength of the reaction, which is suitable for the activity of enzyme. Okay, so suppose this DNA polymerase is having certain optimal pH and optimal temperature, optimal pH and optimal ionic strength to do in a proper way. So that kind of environment is provided by the buffered solution. Okay, so these six are the basic component for any kind of PCR reaction. Okay, so here this is the template DNA. Okay, which you wanted to amplify. Okay, so and this the complementary primers also is there. Then you require one DNA polymerase, that is tag. Okay, either there are basically in routine lab practice we generally use tag and PFU. Okay, so this tag is nothing but it is the thermoaquaticus, and this PFU is fusion polymerase. Okay, so these two enzymes are thermostable. Okay, but the difference between these two enzymes is tag polymerase is nothing having the proofreading activity, whereas this PFU is having the proofreading activity. Okay, so when you wanted to do any sensitive kind of experiment like mutational study, then you always go for the PFU. Okay, and if you just wanted to have an idea like your result is positive or negative, then you use tag polymerase. Okay, the difference between these two enzymes is one is having proofreading activity and another is having no proofreading activity. And then you are having the DNTPs, this individual DNTPs. Okay, and you mix all this component and keep on the PCR reaction. Then for any successful PCR reaction, firstly your primer is the major concern. Okay, while designing the primer there should be certain consideration you have to consider. First one is the primers are short single stranded DNA molecule. Okay, it is around 15 to 40 base pair long. Okay, so this 40 base pair is too high, you require around 15 to suppose 30 base pair. Okay, 30 base pair. Then it once you design the primer, you will give it to any company, and this company will give you the synthesized primer. Okay, whatever sequence you have given, based on that the company will synthesize that particular primer. Then primer are sequence specific. They will bind to a particular sequence in a genome. Okay, and as you design primer with a longer length, like means go beyond the 40 base pair, the primer become more selective. Okay, and then DNA polymerase require primer to initiate the replication. This is the purpose for using PCR primer, is because it is like it is initiating the replication process. Okay, so yeah, it is initial point for replication, you can say. Then um, there is different things we have to consider while designing the primer. First one is the primer length, second one is the melting temperature, third one is the specificity, fourth one is the GC content, and fifth one is the 3 days end. Okay, so primer length, as we earlier discussed, like this primer we have to use generally 20 to 30 base length. Okay, and if it is higher than that, it will be more selective or more specific. Okay, and the second thing is primer length is proportional to the annealing efficiency. Okay, so if you are having a longer primer, then the annealing will be inefficient. Okay, and because you require more temperature for annealing. Okay, if your uh, primer length is high. Okay, then the primer should not be very short, as otherwise what will happen is, suppose this is your DNA, if your primer is too short, then it will randomly bind anywhere. Okay, because your primer is made up of four type of nucleotide. Okay, either A, T, C, G, like that. Okay, so suppose it is the sequence of A. Okay, if your primer is sequence of A, so in this much long template, means more time the A will come. Okay, and it will go and randomly bind with the um, template wherever it will see the A sequence. Okay, like that, if you are having very long uh, um, primer, then it will be more selective. Okay, it will be more selective, but that much specificity we also doesn't require, because if we will increase the primer length, it will create the problem in annealing. Okay, so that is the one important thing we have to consider. The primer length should be around 20 to 30 base pair in length. Okay, then you require the GC content. So ideally a primer should have a near random mix of nucleotide, that is GC content should be always 50 to okay 70%. 50 to 70% GC content should be there. Okay, so and there should be no poly G and poly C stretch that can promote the non-specific annealing. Okay, so it should be the GC content should be 50 to 70%, but at the same time it should not be like the sequence is G G G G G G or C C C, like that. Okay, so there should not be any poly G and poly C stretches, so that it can promote the non-specific annealing. That you have to remember. The third thing is 3 days in sequence. Okay, so that 3 days terminal position in PCR primer is essential for the control of mispriming. Inclusion of a GC residue at the 3 days end of primer helps to ensure correct binding. Okay, so the 3 days end of the primer should be ended with either G or either C, because this G and C is giving 3 hydrogen bonds, so it provides some kind of a strength while annealing. Okay, but at the same time it should not be like poly G and poly C stretches should be there in the 3 days end. Okay, it should be ended with G or C either, but not be in a stretch form. Okay, then you are having the melting temperature. So the goal should be design a primer with an annealing temperature of at least 50 degrees Celsius. Okay, it should be around 50 to below 72 degrees Celsius, because at 72 degrees Celsius the polymerization is happen. Okay, so the annealing temperature always better to at least 50 degrees Celsius and below 5 degree of this 72 degrees Celsius. Okay, um, so it will be better primer. Okay, so that also you have to remember. So this is the procedure. Like in the first step there will be denaturation of the DNA. Okay, suppose this is your template DNA. Okay, and in higher temperature around 95 degrees Celsius temperature, it is separating. Okay, it is separated into two strands. Okay, then in the second step annealing of the primer will happen. Okay, or primer binding will happen. Okay, on a particular temperature like 50 degrees Celsius we have seen like earlier. So 50 to around less than 72 degrees Celsius this primer annealing will happen. Okay, so primer will go and form the complementarity to the template DNA, and yeah, so primer bind to the complementary sequence on the target DNA. Then primer are chosen such that one is complementary to one strand and other one of the target sequence, and other is complementary to the other strand of the target sequence. Okay, and here if you will see like this is the sequence level annealing. Here we can see the template is this one. Okay, and this is the primer, the short stretch of oligonucleotide RNA is primer. So this primer is complementary to this one. Okay, like that this primer is complementary to this sequence. Okay, so it is always complementary to one of the strand of target DNA sequence. Then the extension of primer will happen through the polymerase. The now the using the DNTPs, this polymerase will extend this primer. Okay, when until the template is there. Okay, so till here the and means extension will happen. Okay, so this is the overall procedure. Like in around higher temperature this template DNA will separate. Okay, and each strand will work as a template for next round of the cycle. Okay, and then the second step the primer will come and bind here. Okay, through the hydrogen bonding it will do the base pairing, and then on the 72 degrees Celsius the polymerization will happen. The nucleotide will come and through the help of this DNA polymerase it will extend the primer. Okay, to form the nanced DNA strand. Okay, so like that if you will see here in the first cycle there will be 4 copies. Okay, because it is the 2 to the power 2. Okay, and in the second cycle it is 2 to the power 3, so 8 copies will generate. Okay, so every time if suppose someone asked you like at 2 to the power 31. Okay, means on the 30th cycle how many DNA will be produced, so it will be around 2 billion copies. Okay, so on the 30th cycle it is 2 to the power 31. So you always take n is the always 1 plus then the cycle number. Okay, so suppose here you are talking about the 30th cycle, so you here you will take the 31. Okay, so 2 to the power 31 equal to 2 billion. So in this question it is asking the DNA sequence shown below is to be amplified by PCR. Okay, so there is 4 primers has been given, and in the question it is asking like which pair of primer can be used for the amplification of this template DNA. Okay, so as we earlier discussed the primer is complementary to each of the means each strand of the template DNA or the target sequence. Okay, so firstly we will see like which of the primer sequences complementary to this template DNA. Okay, so here the main thing you have to always remember is like if in the question if you will see here the primer is given from 5 days to 3 days direction. Okay, all the primers is given 5 days to 3 days direction. So if you will consider this sequence, okay, which is already present in the 5 days to 3 days direction. Okay, this sequence and this sequence, okay, which is already present in the 5 days to 3 days direction. This one is also 5 days to 3 days direction. So this won't be complementary to any of the target sequence, okay, because it is also started from the 5 days to 3 days direction. So to making it complementary, you should always target this 3 days end, okay, this 3 days end, so that only when primer will bind to this template, then it will start from the 5 days end, okay, here and here, okay, this is the 5 days end, okay. This side here your free OH group is present, okay. From here the DNA polymerase will amplify the template DNA, okay. So you firstly you have to then you have to check the complementarity of this 3 days into 5 days in template DNA sequence, okay. So here it is the CG, okay. So firstly check in the given 4 option which is providing the CG complementarity. So here it should come either G, okay, then here it should become C, okay, GC, then here it should be T, okay. So like that. So here if you will see C, GCT is there, okay, then here TT, okay. So the complementarity of TT will be A, okay. So this will be the correct sequence, okay. And remaining that if you will see, yes, here also GCTA, GAT, CTG, okay. So this option B and D first primer is complementarity to this side, okay. So we have to check the another side also, okay, because B and D both is having GCTA means complementary sequence of this 3 days end, okay. So we have to again check with another 3 days end, okay. So here the sequence will come G, then here this sequence will come C, okay, G, C, G, C, GC, GC, T, okay. So where is GC, GC, T? Here GC, GC, T, okay. But here it is started from the T, okay. So C is not complementarity with the T, okay. So this will be the wrong option, okay. So option B will be the correct one, because here both the 3 days end is complementarity with these primers, these two set of the primers, okay. So yeah, that's all for today's class. Thank you.